right, so now we're going to look at chapter 6. This is going to look at microbial growth. So we're going to look at the physical growth requirements for bacteria. Now, one thing to note about this is it's actually the number of bacteria, not the size. So when we're talking about growth here, it's increasing the number of bacteria. Now, there's a number of things to look at that can affect the growth. Temperature is one, pH, and also osmotic pressure. So we're going to start with the temperature first. So with temperature, all bacteria have an optimal temperature. They have a temperature that they grow best at. This is a temperature which the bacteria has the shortest generation time, so where it can reproduce very, very quickly. So it's, in other words, it's when it grows best. There are 3 basic categories based on preferred temperature range. There are a few more categories, but these are the 3 we're going to focus in on. We have the thermophils. The thermophils, if you'll notice, like the warmer temperatures. You have the mesophils, which are going to be more of the middle range. And then you have what we call the psychrophils, which like colder temperatures. The range spread for each of these is approximately 30 degrees, and growth is not optimal at the ends of the ranges. So if you see here in the graphs, you'll see at the 2 ends, their growth is very limited. However, when you get towards the middle, their growth increases greatly. All right, so this shows you what they prefer, but they could still grow at those extremes. It's just not optimal. So let's talk about the psychrophils first. These are the cold-loving bacteria. These guys are the ones that like the bottom of the Arctic or the Antarctic Ocean, so where it's very, very cold. So we don't really see much of those when we talk about them being pathogenic. However, there are what we call psychrotrophs. Psychrotrophs are those who like cooler temperatures, just not the really cold temperatures. These guys actually like refrigerator temperatures, and they can cause food spoilage in your refrigerator. They can also, though, grow at room temperature. So they'll grow at room temperature, which is approximately 25 degrees Celsius, and they can continue to grow in the colder temperatures. They can actually grow down to 0 degrees Celsius. So if you look here at the thermometer, it shows you they're below this main kind of danger zone area, but this is when many bacteria survive. Some can't grow here, but they can at least survive. The refrigerator temperatures may allow slow growth of spoilage bacteria. Very few, though, pathogens can grow in this. Listeria is an example here. Listeria prefers temperatures between 30 degrees Celsius and 47 degrees Celsius, but it can grow in a range from 1 degrees to 45 degrees. Okay, so it has a really large range, even though it prefers between 30 and 47. It can still grow as low as at 1 degree. This group is where most pathogens are located. These are the mesophils. They are the moderate temperature loving. They grow best between the temperatures of 25 and 40 degrees Celsius. Most bacteria are mesophils, and again, most of the pathogenic ones are located here. And one thing that is key is our body temperature is 37 degrees Celsius, which is right there in their range. This allows for rapid growth of these bacteria, and some of these actually do produce toxins in this growth range as well. This is the most common food spoilage organism, are going to be mesophils. The next are the thermophils. The thermophils are the heat-loving. These are found in like hot springs. It can also contaminate dairy products, and the reason why they will stay in the dairy products is when the dairy products have been pasteurized and they go to that extreme heat. These guys don't mind that heat, and so because of that, they can still survive. They don't grow well at your body temperature, so a lot of times they're not pathogenic, all right, because your body temperature is actually too low, because if you notice, their low end is right there at 40 degrees and body temperature is at 37. So the temperatures in this range can destroy most microbes when we talk about the thermophils. However, these are the ones that do prefer those higher temperatures. With food spoilage, this is not a public health issue with this group because, again, they don't like to grow in the host or the body, with it being at around 37 degrees Celsius. The next we want to look at is pH. So when we're looking at pH, most bacteria have an optimal pH between 6.5 and 7.5, which is pretty neutral. 7 is neutral. Yeasts and molds, which are part of the fungus group, they actually prefer it to be a little more acidic, so they like the pH of 5 to 6. There is also a group of bacteria that really like acidic conditions, and these are called acidophils. These are found in the group Archaea. They can tolerate really acidic conditions, but again, if they're in the group Archaea, they're not pathogenic. So acidity, anything below pH of 4 is used for preservation of food. This is why we use like vinegar and things like that to pickle things. Like when we talk about cucumbers and making them into pickles, because it helps stop growth of bacteria. A lot of times they need buffers, and we need buffers in artificial media because during metabolism, actually breaking down the glucose into ATP, acids are produced, and if the acids are not taken care of by the buffers, it will start to decrease the pH, which would ultimately affect the growth of the bacteria. Helicobacter pylori is one that is associated with causing gastric ulcers, which are found in the stomach. A lot of times people would think, well, it's found in the stomach and there's lots of stomach acid, therefore it must be an acidophile, and it's not. It likes to gain access into the layer of the mucosa of the stomach, where the mucus helps protect it against the stomach acid. All right, so it's not actually an acidophile. However, it can survive in that acidic condition as long as it can gain access to that mucus layer. The next thing we want to look at is osmotic pressure. Cells are composed of mostly 80 to 90% water. So the water content around them is very important because we talked about in a previous chapter the movement of water. Remember that water likes to move from po to per. And so if conditions are altered or different from what the inside of the cell is, water will either move into the cell or out of the cell. So when we look at hypertonic environments, these can cause plasmolysis or cell shrinkage because, again, the water will leave the cell. There are a group who actually like hypertonic environments. These are called haliophils. Again, they're part of the Archaea group, and they're found in like the Dead Sea and the Great Salt Lakes because they're salt loving. And this is going to be bacteria that's even more salt loving than what the ocean has. The percentage of salt is a lot higher. So this again is why we use salt a lot of times as a food preservative, because it will cause a hypertonic environment which the bacteria cannot survive in. Agar is 1.5% higher concentration, which cause hyperosmotic conditions sometimes. And so when we look here, plasmolysis inhibits the cell growth, so it doesn't allow it to grow too quickly. Some bacteria are what we call heliotolerant, okay, meaning that they can grow in that condition. They tolerate the salt. It's not their favorite, it's not their optimal, but they can tolerate it. This is known as facultative because they can grow in those particular environments. One of the main examples here is Staph aureus. This can be used to help identify certain organisms versus others, because if you'll notice, some don't grow as well on the plate, like you see here in the blue, versus the one that grows better in that haliotolerant condition, okay, what you see with the orange arrow. We also see hypotonic environments. This is where water moves into the cells. The cells will start to swell, and this can cause osmotic lysis, causing them to swell to where they could potentially burst. All right, and so that environment's not conducive for bacterial growth either. So based on these conditions, what factors are used to help reduce food spoilage? Because again, we want our food to last as long as possible. Well, of course, we can do refrigeration. Refrigeration can stunt the growth of some microorganisms, even though it doesn't kill them. We also could use acids because the acidic pH causes them to not want to grow, and this is why we might use vinegar with the whole idea of pickling. And then also adding salt. Especially before refrigeration was an option, salt was the main way to help preserve food. All right, so that was the physical requirements. Now let's look at the chemical requirements for microbial growth. When we look at microorganisms, just like any other cell, they do need a carbon source. When we're talking about the chemioheterotrophs, they need a way to be able to access carbon. This is going to be through organic sources. These organic sources are going to be those macromolecules we discussed in previous chapters, like carbohydrates, proteins, and lipids, which are fats. We also see that bacteria and other microbes are also going to need nitrogen. This is required for them to make proteins as well as nucleic acids, which helps with their DNA and RNA, as well as helping to synthesize ATP. So if you look here in the proteins, you can see that nitrogen is present. This is the amino acid monomer. We also see with nucleic acids how important the nitrogen is. You can see how many N's are located here for the nitrogenous bases. And also for the ATP, there's the nitrogen side due to the nitrogenous base as well. Another chemical component that's needed by the cell is sulfur. So you'll notice here are some examples of some proteins where the amino acid contains the sulfur. This is the R group that makes each of them a little bit different based on what components are made up in that reference or R group. We also see that sulfur is important for some vitamins, like in this case, vitamin B1. This is a precursor to a coenzyme that helps metabolize pyruvate. All right, and so this is going to be an important precursor to help those enzymes function properly. Another chemical component that is also important is phosphorus. Phosphorus is found in nucleic acids because nucleic acids have a phosphate group. We also see it's important in ATP, the fact that it is adenosine triphosphate. There are 3 phosphate groups here for ATP. And the phospholipids. Those phospholipids have a phosphate group present, and this is what makes up the cell membrane of the organism. Now, those were all the major elements that are really important, but there are some trace elements that are needed in small amounts. A lot of times these are present in tap water, so just using tap water or having that kind of water around gives them enough of these trace elements to help. The typical role of trace elements is for cofactors. These are going to be like your irons, your coppers, zincs, things like that, which again can be found in normally water, and they are going to help enzymes work properly as cofactors. Now, other essential organic growth factors are going to be important, not provided by their diet, but they are needed to help with coenzymes. So an example of this is Haemophilus influenza. It's what we call a fastidious bacteria, which means it's picky. Okay, it's going to need some extra help. And with this, there's some factors that are present that are essential for its growth. One is what we see here with the Roman numeral 10 or X. This is what we call Hemin, and then also the V or 5 Roman numeral is NAD. These are growth factors required for metabolism to have to be added to the growth media. So if we just take a normal growth media like you see here, there's no growth. But if you add to the growth media these 2 factors, you do see that this bacteria can grow there. However, it's very picky, meaning it has to have those factors present in order to grow. Another thing that could be important for the growth of bacteria is oxygen. Now, this is whether or not it needs oxygen or it cannot be around oxygen. All right, so let's look at this table and we're going to break each of these down. So this first group is the obligate aerobes. Okay, these grow well on media exposed to the air. Thus, you can see in the test tube, they're all growing at the surface where oxygen is present. They require oxygen as their terminal electron acceptor. Therefore, they have to have that oxygen there to create the water. Some of these will test positive for that cytochrome C with the oxidase test because they have the electron transport system. All right, so this is the obligate aerobes. They have to have oxygen. The next ones are the facultative anaerobes. These can do both aerobic and anaerobic growth. They have a greater growth when there is oxygen present, so they can grow with or without oxygen. However, if you'll notice at the very top here where oxygen is present, their growth is a lot more dense because it is better. However, they can spread out throughout the media where oxygen is not located. They can switch back and forth between aerobic respiration and anaerobic respiration. So they can generate ATP either way. One of the best examples here is Escherichia coli or E. coli. The next are the obligate anaerobes. These are obligated to not be around oxygen in order to grow. Therefore, they cannot use oxygen as their final electron acceptor. One of the main groups here are the Clostridium group. They lack enzymes to detoxify the effects of oxygen. Therefore, oxygen is like a toxic gas to them. If an obligate anaerobe is to be isolated, you have to do special collection and transportation required in order to grow this, because if it's exposed to oxygen for an extended period of time, it does cause these bacteria to not grow. And you'll notice that they're growing at the very bottom of the test tube where oxygen is not available. The next group are the aerotolerant anaerobes. They're only anaerobic growth, but they can actually continue to grow in the presence of oxygen. So they will only be anaerobic, but oxygen doesn't bother them. They cannot use the oxygen as their final electron acceptor, but they can tolerate it being present in their area. They do produce enzymes that help detoxify the effects of that oxygen. The last group are the microaerophils. These are going to do aerobic growth, but they need very low concentrations of oxygen. So oxygen is required in only very low amounts. And again, this is why you can see in the test tube they're growing right in the middle, where there's some oxygen present, just not very much. So now that we know the physical and the chemical requirements for bacterial growth, this can lead us to actual culture media. This is what we can end up using as a substance to grow bacteria. This could be a broth, which we see as a liquid, or it could be an agar. And now agar is going to have thickening agents in it to make it more of like a gel substance. Now, some bacteria can grow on any type of media. They're very they're not picky. They can grow on pretty much anything, but some actually need special media in order to grow. Now, when we look at this broth or agar, we can put it in tubes. All right, so you can see here in the picture, you have the broth, which is the liquid in the tube. We can also have it as a slant. This is agar that they've put in the test tube, and then they have the test tube as it solidifies into the agar, and they do it at a angle, so it makes a slant. Or we can even do a deep. And again, this is agar, but it has been poured and it stays straight up, creating a deep. We also could use petri dishes. You can see them here. These are agars of petri dishes. The petri dishes are just going to give them a nice field of media to grow on. One thing to note about this is if you ever work with these in the lab, make sure that you label the actual bottom part of the agar plate because the lids can come off and there's no agar on them. And so if that comes off and they get mixed up and stuff like that, you wouldn't know what was growing on the plate. So you want to make sure you do it on the bottom side that has the agar attached to it. Now, when we do these, whether we're doing a tubed type of media or a petri dish, we want to make sure that that media is sterile prior to its inoculation. We want to make sure that there are no endospores present and also no vegetative elements. Okay, something's not already growing in it. And so we want to make sure that it's sterilized before we pour it into these containers. All right, this ensures that whatever we culture on it is what we're really wanting to grow, that it wasn't contaminated before you inoculated it. So inoculum is what is placed in or on the media to start growth. Okay, so a lot of times we'd say we would use an inoculating loop in the lab. We would sterilize it, collect the sample of what we want, and place it into the broth or on the petri dish. This will be what we are wanting to grow. When it grows, this is called the culture. This is the bacterial growth that is in or on the media. So if you see here in the broth, you can see that at the top, before it was inoculated, it's clear. It's going to be cloudy. On the other hand, you can see here when it talks about the petri dish, you can see that there's obviously lots of different colonies or different types of bacteria growing on this plate. So some properties of agar we want to look at. When we're dealing with agar, it does become a liquid at 100 degrees Celsius. It does gel at about 40 degrees Celsius. We can pour it in tubes or plates, and depending on how we lay the tube will depend on if we have a slant or a deep. A lot of times we can also add nutrients to this media in order, or this agar, to supplement the bacterial growth. This could help us actually differentiate between types of bacteria or make sure that the elements are there for that bacteria to grow. So when we look at these nutrients in the agar, we do need to make sure that certain things are present, and this includes a source of carbon, nitrogen, sulfur, and phosphorus. Those have to be present for all bacteria. Now for others, if they're fastidious, meaning they're picky, we may have to add other factors to it in order for them to grow. So this is just a picture. This can come out of like any kind of, if you do a web search of culture media, you can find a picture like this that shows you lots of different types of media. These are various nutrient agars and broths that can be used for bacterial cultures. Some of them have different colors for a reason because they have different components in them. They also may change color depending on what's growing on them to give us an indication of what's present. And so you're going to be learning a little bit more about some of these in more detail in the lab section. But let's take a look at a few of them. So we can look at what we call chemically defined versus complex media. These are considered non-selective media because they support growth of most bacteria that are not fastidious or picky. When we look at chemically defined medium, we know exactly the chemical identity of all the ingredients. So we know exactly what's in it and we know what amounts are in it. All right, so if you'll notice here on this label, it's telling you specifically what's in each of these. However, when we talk about complex media. They do contain substances and it tells us what's in it, but the exact amount of a particular chemical is unknown. So if you look here where it says peptone, it says partially digested proteins. We don't know what kind of proteins were in there. We weren't specific of what type. Same thing with beef extract. All right, that's just mixed in there. It also just tells you that it's 15 grams of agar and 1 liter of water. It doesn't give us specific amounts of these different parts. All right, and so because of this, so the contents of this could vary batch to batch. Other media types can do some different things here. We see that some of them are reduced media, and this means that they can actually allow the growth of obligate anaerobes, meaning that the ones that cannot be around oxygen. We see some can be selective. This is where they'll inhibit some bacteria but encourage the growth of others. So they're selecting what gets to grow on it. Others can be considered differential. This is going to be where we can distinguish between bacteria based on their colony type. All right, so letting us know too, like by color changes or how they look when they're growing on that type of media. We also see that there can be enrichment media, which promotes growth of pathogens in low numbers, and it inhibits other bacteria that's actually not a problem. This is really important for fecal cultures because you have a lot of bacteria in your intestines that actually helps you. It's beneficial to have there. We don't want that to grow and not allow the pathogen to grow if we're trying to figure out what is making you sick. So with that, we want to make sure that it's an enriched type of media that allows the pathogens to grow and not the good bacteria that you already have in your digestive system. All right, and so you can see the purposes behind each of these types of culture media listed here. All right, so we're going to kind of look at just some groups of these different types of media. Or in this case, these are all types of blood agar, meaning that they have some type of blood present in the agar, but you'll notice they all look a little bit different. This first picture is a nutrient agar with 5% sheep blood. This means that it has whole blood cells in it. All right, and so this is known as BAP. This is a non-selective media. So this is a differential media in the sense that it will let you know if the cells can do hemolysis, meaning breaking down the red blood cells that are present. So you can see here there's the alpha, the beta, and the gamma that you can see here in the plate. On the other hand, the middle picture here is of chocolate agar, also known as CAP. This is late sheep blood, meaning that the blood has been the blood cells have ruptured and the contents are out, and so they don't have to access the whole cell by itself. So this is considered a non-selective media. So the last one over here is the Thermartin or TM. This is CAP or chocolate agar, but they've also added antibiotics to it. Therefore, it's going to be very selective. It's going to do selective isolates. And so one example of this is it helps isolate Neisseria gonorrhoeae. All right, another example is the eosin methylene blue or EMB. This is going to be selective for gram-negative bacteria, therefore it's differential. You can see also that some are lactose negative versus lactose positive. They can give that more metallic type of green color or metallic color when they are lactose positive. Okay, so it's going to be selective for gram negative, but it also is going to be differential for whether they're a lactose fermenter. The next one is MacConkey's. MacConkey's is also known as MAC or MAC. This is also selective for gram-negative bacteria, so it does not allow gram-positives to grow, and it is also differential where you can see the pink color versus the yellow color. We also have hectonic enteric agar or HE. It's normally a greenish clear color. However, it is selective for gram-negative bacteria and it's differential. It does allow for like Shigella and Salmonella to grow on it. We also have a phenylethyl alcohol agar or PEA. This is selective for gram-positive. So you can see the gram-positive here versus the gram-negative. We also have the mannitol salt agar or MSA. This is selective for gram-positives that are haliotolerant, meaning they can tolerate the salt content, and it is also differential. So if you look here, you can see that both these are different species of Staphylococcus, but the color difference shows you which one they can differentiate against. Both can grow on this agar, however, the color lets you be able to see that it is also differential. Another thing we want to note is that incubation can be different with these different types of bacteria. Some need incubation for what we call capnophils and obligate anaerobes. Capnophils grow better in a higher level of carbon dioxide. So typically all the culture plates and so on are placed in what we call a CO2 incubator. Now, of course, there are these more elaborate CO2 incubators that could be used, but in a lot of labs, what they do is they just take a jar, place the petri dishes and tubes inside, and light a candle. The candle will be able to burn until all the oxygen is pretty much used up, and the CO2 is what's left behind. We also see that there's things that are anaerobic chambers. These are for incubation of reduced media, and remember, they also need special conditions because they can't be around oxygen. The ability to grow bacteria is only as good as the specimen that's collected and how it is transported. The selection of the following is really important whenever you collect something and you transport it. And one is the appropriate media, making sure you're using the right type in order to grow with what you need. Also, the environment to incubate it in. Is the temperature set towards optimal? Does it need carbon dioxide? Is it anaerobic? Also, the length of time. Some of them can actually grow very quickly, where others take more time, so you're not going to see results as quickly. Some bacteria cannot be cultured on artificial media. We find that on the artificial media, it's not able to be cultured. And one of these examples is syphilis, which is a spirochete. Another is the Mycobacteria leprae. We see that it cannot be cultured on a plate or in a tube. And the last are the obligate intracellular parasites like rickettsia and chlamydia. Now, there are a couple of different ways that we can inoculate plates. One way we want to look at is what we call for obtaining pure cultures, to where you're obtaining one particular type of bacteria. This is called the streak plate method. What we're looking at is we're trying to isolate colonies. Colonies are known as the CFU, or colony forming unit. A lot of times they start from one particular bacteria and then grow into a colony. When we look here, the culture media is inoculated, normally under a flow hood. We want to see that we are isolate colonies to where they're all by themselves. And so in this top picture up here, you can say that there's good colony isolation, but down in the bottom, there's poor. There's too many colonies close together to be able to isolate that is one particular colony. The way that you do a streak plate method is that you take your broth or whatever you're taking your sample from, and you place it in your your field number 1, and you streak it, normally using a sterilized Q-tip. Then you're going to take your inoculating loop, you're going to sterilize it. Once it is cooled, you're going to streak across it like you see here in number 1. You're then going to sterilize the loop again. When you sterilize the loop again, let it cool, and you're going to drag a little bit from area 1, and you're going to then go and streak through area 2. Again, sterilizing the needle or the loop. Once it's been sterilized and it's cooled, you do the same thing where you drag through 2 into 3, and then finally 3 into 4. Hopefully then you've diluted it enough to where you get single colonies by the time you get to area 4. So how does this bacteria grow? When we're looking here, remember, we're looking at its growth as in not its size, but in its number. So this is actually looking more at reproduction of bacterial cultures. The reproduction of a bacteria is called binary fission because it does not contain a nucleus. This is where one parent cell is going to divide and yield us 2 daughter cells. These daughter cells are going to be genetically the same. This process starts with, of course, replicating the DNA. Remember that they have a single circular strand of DNA, and they are going to then replicate it. Once they are replicated, the cell is going to start to elongate. It's going to get longer. As it gets longer, it's going to start to divide in the center. This is going to allow the plasma membrane to grow inward, as well as the formation of the cell wall, until we finally have 2 distinct cells. All right, and so we go from having the 1 cell to having 2 distinct cells. All right, and so this is the process of binary fission. So how long does it take a cell to do this? This is known as the generation time. This varies per organism. Some examples are E. coli can actually reproduce every 20 minutes. So we can go from 1 cell to 2 in 20 minutes, 2 to 4 in another 20 minutes, and 4 to 8. So within an hour, you can go from having 1 cell to having 8 cells. Okay. On the other hand, when we look here at M. tuberculosis, this one takes 18 hours to go from one cell to two. So you're obviously going to have to incubate this one longer in order to actually see results of these colonies. We do see that this is logarithmic, but there are some limitations. All right. And so when we see this, there is that logarithmic dividing where we see that you go from one cell to two to four, and it just keeps increasing like that. However, there are limitations because this is based on the space as well as the nutrients that are present. Once they start to run out of nutrients, it's not able to continue to divide. The gestational time determines the culture time and also the incubation of diseases. How long does it take when you have been exposed before you start seeing actual signs and symptoms of you being sick? Because you have to have enough time for that organism to replicate in order for you to see that you are now sick. Okay, so if you take a look here, this shows you that after 20 generations, there are over 1 million of these cells. So they can actually grow very, very quickly. So let's look at the phases of growth when we're looking at it in vitro. So this is looking at the growth in vitro, meaning it's in a petri dish or it's in a broth tube or something like that. It's not in vivo inside of a body or cells. So we see that the first phase here is called the lag phase. This is a few hours after inoculation. The cells are preparing for their growth, all right? So they're making sure that they can replicate their DNA and they start that process. There's no increase in the population size during this. Okay, so no in vivo, like inside of a host, this is also known as the incubation period or the time after exposure. This is before symptoms will begin. So a lot of times we'd say the person is asymptomatic at this point. Then we move into the second phase. This is known as the log phase. This is where we actually have logarithmic or exponential growth. And you can see that in the chart that it has a huge spike in the growth of the population. The number of organisms can double every generation time. The best time to perform biochemical testing is during this time because all of the cells are very metabolically active. And a lot of these tests are looking at those metabolic components in order to create positive or negative tests. Note, in vivo, this is when symptoms of the disease would start to appear. So this is where the person, if they have contracted the disease, start to show some of those symptoms, whether it's a fever, a cough, runny nose, things like that. We then have the stationary phase. This is a stabilized phase, and you can see that it is a plateaued group. This is a period of equilibrium. So the death rate of these bacteria dying is equal to the growth rate. So you don't see any extra growth. It's equal here. Nutrients are starting to deplete, and waste products are starting to accumulate. And so because of this, it's causing it to have a more stationary phase. In vivo, this is when the immune system is attacking the bacteria and hopefully getting an advantage over it. The last phase is known as the death phase. This is where the population size is decreasing, but it's decreasing, if you'll notice, at a logarithmic rate. It's going down, all right? And it's going down pretty steadily. The nutrients become more and more depleted, and the death rate now is way greater than the growth rate. In vivo, this is when symptoms start to disappear, and we call this the convalescence time that begins. Now, a person can still expose somebody else and sometimes pass it on to somebody else, but they don't actually have real true signs and symptoms of the disease anymore. So the next thing we want to look at are the methods of measuring microbial growth. How can we actually measure how much growth is present? One way is called a direct way, or also known as a quantitative, meaning we're actually going to do counting here. These are called plate counts, and this is going to be looking at the CFUs, so the colony forming units. And we can also do this by doing serial dilutions. This is a technique used on food and environmental specimens to determine the level of bacterial contamination. All right, so when we're talking about like the big thing that you may see is, oh, they had a salmonella outbreak with this and they had to pull it off the market to see how much is there, or listeria, or E. coli. What they're going to then do is take these samples and they're going to do some serial dilutions and actually start counting the CFUs, doing plate counts. So we're going to take the original inoculum and you're going to place 1 milliliter of it into your broth. So you were diluting diluting it 1 to 10. 9 milliliters of broth in each tube, and we're going to put 1 ml of the substance. We then plate it out. We then can take 1 ml of that dilution and then put it in the next one. This is now a 1 to 100 dilution. We do it again to a 1 to 1,000, 1 to 10,000, and then 1 to 100,000. And if you'll notice as we plate these out, the growth starts to get less and less. Okay, we then though have to count how many are present there and multiply it by the number of CFUs by the dilution factor. So if we go over here to the very end that we have in this example, there are 3 colonies present. We would have to do 3 times 100,000. This means that there are 300,000 colonies in that original type of specimen. This is usually reported in the number of the colony forming units per milliliter. All right, so this one would be that there would be 300,000 colony forming units per milliliter of this substance. In the lab, you will be using directly measuring and quantitatively bacterial growth for urine cultures. So when you get to the lab where you're going to look at urine cultures, this is what they look at. So you look here that it's 10 to the negative 3, or it's a 1 to 1,000 of a milliliter of this specimen plated. So again, you would count how many are there and multiply it by the 1,000. A lot of times they'll do this by using a calibrated loop so that you know exactly how much fluid that loop picks up. So when we look here, this particular colony count, if you count the colonies, there are 8 of them there. You do 8 times 1,000, so it's 8,000 colony forming units per milliliter. There's also some indirect ways that we can measure the amount of bacteria. This is known as a semi-quantitative because you're not actually counting colonies. This is a semi-quantitative way where we can use what we call turbidity. How much of the light passes through the substance? Is it a clear solution, or is it clouded up due to bacterial growth? All right, and so a lot of times we can use what we call a spectrophotometer, where we can use light and it then collects how much light comes through. You always have to set it with a blank tube that just has a liquid in it, and then you can put the tubes in that have been inoculated to see how much comes through, the percentage of light. Okay, because this light is scattered when it hits those colonies or those groups of bacteria. We also see that you can look at the McFarland standards. This is a 0.5. It's used by Kirby Bureau antibiotic sensitivity testing, and we will talk a little bit more about this in chapter 20. But if you look here, it's an adjustment of the turbidity in the range to standardized testing. Okay, so what we're doing here is you can see that you can't see the lines through this far one, and it gets less and less. So it shows you that we have less and less bacteria based on that amount, a lot of times, of whichever antibiotic is present in this particular group. In lab, you can also use a semi-quantitative measuring of bacterial growth, and so you don't actually count all the colonies, but you can say whether they have light growth, moderate growth, or heavy growth. Now, in light growth, a lot of times you could be able to count them, but because you don't know the dilution, you're not going to be able to give an actual quantitative measurement. On the other hand, on the heavy growth, it's going to be very hard to tell how many are actually present there. All right, so this covers chapter 6. Again, if you have any questions or concerns, please let us know.